Namaskar, welcome to PK's classes. Today we are going to study the mechanism of fluoroquinolone resistance. Before that, let us discuss how fluoroquinolones act. As you know, the DNA first there will be unwinding, and then from one DNA we can get two DNA. But for this replication process, the DNA has to be in relaxed state. In bacteria, this DNA is present as the circular one, and this circular one in relaxed state it is required for formation of two DNA, and initially when there is formed, there is catenated, then they are separated into two separate DNA, and this catenation, this is catalyzed by topoisomerase 4, and usually this DNA is in condensed form or supercoiled form, so this circular DNA is in the supercoiled form, and supercoiled form has to be converted to relaxed form, then only it will undergo replication and two DNAs will be formed from one DNA. So the supercoiled form is converted to relaxed form by the action of DNA gyrase or topoisomerase 2. So you can see the supercoiled form is converted to the relaxed form by topoisomerase 2 or DNA gyrase. Then the catenated form or knot form are converted to the relaxed form by topoisomerase 4. So these two enzymes are required for DNA replication, and fluoroquinolones inhibit these two enzymes. Fluoroquinolones inhibit DNA gyrase and fluoroquinolones inhibit topoisomerase 4. So the mechanism of resistance, there can be alteration of the drug target enzyme, that is either DNA gyrase or the topoisomerase 4, and this DNA gyrase consists of four parts. DNA gyrase, and so DNA gyrase has four parts. We can see that. You can refer to our earlier classes on mechanism. There are two gyrase A subunits and two gyrase B subunits. So the two genes are gyrase A and gyrase GYR-A, GYR-B genes. So mutation of these two genes can alter the target enzyme, that is DNA gyrase, and most common is gyrase A gene, and this is located in the quinoline resistance determining region or QRDR, and mostly in case of gram-negative bacteria. Similarly, the topoisomerase 4 enzyme consists of four parts, 2 par C and 2 par E, and here also the mutation and two genes are there, par C gene and par E gene, and mutation in par C gene, mostly in case of gram-positive bacteria, causes the alteration in the drug target enzyme, so that there will be no binding of the fluoroquinolone with the target enzyme, that is topoisomerase 4. And gram-positive bacteria like Staphylococcus aureus, Streptococcus pneumoniae, they show such type of resistance. Then the second mechanism of resistance is alterations in access to cytoplasmic drug targets. So this can be by reducing the drug diffusion into the cell, so into the bacterial cell there will be no drug diffusion, so that there will be less drug available. So this occurs because of the decrease in level of the outer membrane proteins called porins. So there are structures called porins which help in entry of the fluoroquinolone into the cell. So if there is modification of these porins, then there will be no entry of the drug into the bacterial cell, so that will cause resistance. Second one is the MDR efflux pump expression, and MDR, you can see this is the porin structure, so this is required for entry into this, and these are the two targets, the DNA gyrase and topoisomerase. So any modification in the porin will inhibit the entry of the drug, that we call as decreased permeability. Then there may be presence of efflux pumps which will throw the fluoroquinolone out of the bacterial cell. So two class of pumps are there. One is major facilitator superfamily pump, MFS, and second is resistance nodulation diffusion, RND family. And in gram positive like Staphylococcus aureus, MFS pumps are there, and in gram negative mostly RND family of efflux pumps are there, which cause the efflux of the fluoroquinolones and hence shows resistance. So the third mechanism can be the plasmid mediated quinolone resistance genes, or quinolone resistance we can say QNR genes, or PMQR, plasmid mediated quinolone resistance, and this occurs mainly in case of Klebsiella pneumoniae. So this resistance causes, this is plasmid mediated, this resistance causes the protection of the quinolone targets. So because of that protection, the fluoroquinolones now cannot quinolone resistance, so protects the quinolone targets from inhibition, and hence the fluoroquinolones cannot bind to the targets, and that produces the resistance. So this is all about the mechanism of fluoroquinolone resistance. Thank you.